Muchas gracias por invitarme. Es un placer estar aquí en el centro de Universidad de Maíz. That's the wrong talk. A ver, bueno. Es un placer ver muchos amigos de Simit y de Colpos y Chapingo. Listo, ok, ¿todo bien? Bueno, yo quiero platicar un poquito. Oh, I'm going to go in English. Like 2 slides in my bad Spanish, and then I'll go to English. No, actually, I'm going to start in English. Let me just do it in English. So, here I just want to talk a little bit about this ear of maize is segregating for a number of major gene effects on things like you have sugary in here, lots of different kernel colors. And the point of that from variation that's observable down to some genes. Over here on the right side, this set of histograms of plant height in a series of maize biparental crosses. And you can see that in every cross, you essentially have a continuous normal type distribution of plant height. So the variability we see in these quantitative traits is very, very different. And what I want to talk about is what have we been able to do in terms of going from these observable complex trait variances down to the gene level. And then if we're able to do that, can we use that sort of information for breeding? Esa es una propaganda. So, estamos escribiendo un libro de genética cuantitativa y análisis de datos. En este momento, si haces un Google search de Ficret, dice $100, algo ridículo. Ahora es gratis. Get it now. Okay. Has nothing great genetic analysis in maize. And I'm remembering I was actually at the colegio. It's a small sample of lines. It's only 26 founders, but it's basically try to maximize as much diversity, global diversity, as we possibly can in this sample. Cross them to B73, and that's important. We use this reference founder so that we can get reasonable adaptation on all the mapping populations of this haplotypic region or this chromosomal region. Then we do selfing for a number of generations until we develop recombinant inbred lines. And what these things represent, so we have 200 lines from each of these crosses, and they represent most of what we've done is that this is all mapped with we have a common set of markers on these things. They're all phenotyped in the same set of environments, so it's much easier than trying to pull together a bunch of distinct biparental mapping populations and try to analyze them. Study, and these are actually small plots, so it's actually a lot of plots crammed into a single field here in Clayton. So it was 10 chromosomes of maize, so these little bars separate the chromosomes. And you're doing just a linkage scan and looking for evidence that there is QTL in a particular region where you allow each family to have a unique effect for the allele. And you can see we get these really crazy, huge LOD scores, and we get very excited. So even at very, very stringent statistical thresholds, we identified many QTL for what we thought was a relatively simple trait, which was flowering time. So something on the order of 40 QTL, depending on where you draw the line. Another important thing we found was even for this, what again we thought was a relatively simple trait, there's very few large effects. The largest allelic effect genes, each of which tends to have small effects. We got excited because you can actually predict, if you take this QTL model and just predict back what the parental phenotypes are, you get a very nice, where region is sort of defined by by linkage, right? So it depends on centimorgans. So you will pick up effects that can be within 5-10 centimorgans of the marker. We want to do something more precise, which was to do a genome-wide association type study where the idea is to get an association test at much higher resolution. In other words, we want to identify the effects of much smaller regions of the genome. And the way we did this is we had sequence data on these founders. So you can think about, we'll get there in a second. Let me start. We have this linkage map data I already told you about. We have something, there was something like originally 1,000, it was like 1,200 markers. On all the progenies, and these things were directly genotyped. So we had real data on all the progenies from these. We had real data on the parents too. So you can track for any, here's a particular parent, and that gets crossed to this B73, and you generate a series of these progenies, you could track in this genome region, we know where this line, which founder allele is in, or haplotype in that region is being inherited. But on the founders, we also have this very high coverage, high dense sequence data where we had millions and millions of these SNP variants called. And what you can do then is just impute with pretty high confidence this information between the linkage markers. You're just basically saying if I have these two linkage alleles here in this line, then I'm going to inherit this block of the founder, and I can look at these individual SNP variants that are in between the things we actually genotyped. So you have, on the one hand, you have this sort of linkage-based kind of QTL mapping with the 1,000 or so markers. You have basically QTL-type resolution where you're probably going to be within 5-10 centimorgans of a causal effect. But this GWAS analysis, you can control for the genetic background for the other QTL you've mapped, but go in a region and look more precisely because the linkage disequilibrium that occurs at a single SNP depends on the linkage disequilibrium in these founders, which is much, much lower than the map type resolution. So that's nice. And so in some cases, we've been able to go from basically observable phenotype variation, map the thing at linkage resolution, so you get down to a few centimorgans, and then get down to the causal gene. Here's one success story. This is this gene, CMCCT, which is the major QTL for photoperiod response in maize. This little graph here these are the 10 chromosomes of maize. Here's 1 through 5, 6 through 10. Here's a linkage map down here. These green boxes are the QTL. And you can see these QTL are kind of big chunks of the genome. These red dots are the GWAS analyses, and the higher up they are, then the more important they are. And you can't see it because I made it so small, but there's one here that's basically has very, very high evidence for a single variant that's associated with this flowering time, photoperiod response. But that, it's still, that's just an association. It's a correlation type study. It required doing very high resolution fine mapping where you control the genetic background and you basically select for recombinations in the region where you've got that QTL. And John Dobley's group did this. And the other thing they had was they had a teosinte allele introgressed into maize. That teosinte allele had a really big effect. So having that big phenotype effect and having high resolution mapping allowed them to identify the causal variation is either in the first exon or in the promoter of this ZMCCT gene. And then independently, a group from China basically did transgenic work to show that if you do a overexpression or underexpression based on what the promoter is of this gene, you can cause this phenotype. So that's a great case of where we went from just the variability in phenotype down to the actual gene. But consider this was a huge amount of work, and we didn't really get the gene directly out of the statistical analysis. It required further work to get there. So here's another case where we're looking at southern leaf blight resistance. All of this is quantitative. There's no immunity. We're not looking at any major genes for resistance. It's basically 1 to 9 type scale. We inoculate these plots, and this is with the colleague Peter Ballankurdy. His people will go through and rate stuff multiple times during the season. It's a huge amount of work. We do a bunch of analysis. You do this genome-wide association study, and then you get a graph like this where, again, just I'm not going to go into the details, but the higher up you are on this plot, the more evidence there is that SNP variant is involved in the variation seen in the phenotype. And what we've done is we put annotations on the gene that these SNPs are in on this thing. So this was exciting. A plant pathologist looks at this and says, oh, there's plausible reasons that some of these things can be involved in quantitative type disease resistance. And focusing on this one here, this caffeine oil CoA O-methyltransferase, the idea was, can we do some functional biological validation? Actually, is not correct. The Peter Ballankurdy and Chin Yang, a postdoc in his group, they basically have done the follow-up validations. And here's just one example of what they've done. So you know what gene you're interested in maize. You can go to a resource called the Uniform Mu Stocks, where the mu transposon has been released in a uniform genetic background. And if you tell them what gene you're interested in, they can tell you which stalks carry insertions either in the gene or near the gene. And there were 2 stalks that had insertions in the 3' untranslated region. we didn't know if that was going to cause any effect or not, but it turns out it did. So if you compare the two stalks that have the insertion at the 3' end of this thing, they actually have higher resistance, so the higher up you are in the scale, the better you are. And if you look at them in the field, you can take pictures like this, and it looks really nice. And what's weird about this is having this transposon insertion in the 3' end actually increases the expression. So this is unpublished. It's not 100% sure, but this looks like a case where, again, we've gone from a complicated phenotype all the way down to a gene. And this phenotype, we had no idea what the biological pathways were in the beginning. The photoperiod response, there's a lot of work in Arabidopsis and rice and things like that where we could rely on saying, yeah, this is a good candidate gene. In this case, we don't really know much of anything about what causes this kind of variation. So it's very handy or very useful to be able to do forward genetics, where you just go from phenotype, you don't know anything about the biology, and just use genetics, get down to some gene, and then you discover something potentially novel. So those are a couple exciting success stories. 10 years of work, millions of US dollars, go into this thing, and we write lots of papers, and I get invited to conferences. I don't know why, because more often than not, what happens is, I don't translate that, but what happens is, and I'll talk a little bit about this later, what we're doing, but when you do these genome-wide association studies, you have to make sure you're accounting for the background polygenic variants. And what happens a lot, I have students who do this a lot, they do the analysis, and once they fit the background polygenic variants term, there's no variability left over to be accounted for by single gene variants. So the polygenic part of the model takes up nearly everything, and there's no power to detect single gene variants. And the student, this just happened a few months ago, I'm in trouble, I'm going to be fired, I'm a disaster because I didn't find a gene, right? So, yeah, you have this problem. Where's my gene? It's genetic, there's genetics, it's heritable, but there's no genes. Where did it go? And, the answer is, well, actually, you have an answer. The answer is that there's lots of genes, and they're very small effects, and you simply don't have power to identify any specific one. And again, thinking about it in terms of breeding, if what we're trying to do is identify genes with large enough effects that are going to be, deployable in a breeding program, you've got your answer. The answer is you don't have it, at least in this sample of germplasm. Okay, it's polygenic. So forget about the idea that you're going to identify a major gene from some, exotic germplasm and just move it right into an adapted variety and you're done. Which was, I confess, my naive idea when I started this. I thought, oh, this is going to work. It's going to be great. I don't think that's going to work. There's some statistical issues. I won't get into it. And I just want to point out this, too, that we publish the ones that work. And a lot of times, we've tried to fine map things like flowering time QTL. And again, flowering time, we can measure it, has extremely high heritability. We put all our genetic, resources into it, and at the end, basically what happens is you start with a QTL, you do fine mapping, you break it down into little regions by recombination, and basically the effect disappears. Where did it go? Well, the effect was probably caused by lots of linked small effect variants, and as soon as you have recombination break those up, your QTL is no longer a QTL. It's a bunch of small effect variants, and that's your answer. And we don't know what they are. But practically it doesn't matter what they are because they're no use. Okay, so there's all this disaster that you never hear about. So I'm here to tell you that it happens. A lot of projects end like the car in the fancy car, a very fancy sports car, and you drive it into a lake. I won't go into that. One other thing that, okay, the QTL analysis, we identified 40 QTL, for example, for flowering time. That looked great. We thought, well, let's take a little more careful look at it, because what can happen when we do those analyses where you put all the families together and you're looking for a QTL that has kind of information across all these families, that's like what we're doing in the bottom of this graph here. I'll tell you what the other part is, but this JF thing here is this thing. So here are 10 chromosomes of maize, and what I'm showing you here is if you take your data set, if you take your data set and you randomly sample 80% of the lines, and you do the QTL analysis, and you say, here's my threshold, here's what I declare as a QTL, and you keep track of that. And then you take another random sample of 80%, and you redo the QTL analysis, and you say, here's what I declare a QTL. And you do this, let's say, 100 times, 80% samples of your data set every time. You ask the question, what for each marker in the genome, that's what these little bars are, how much evidence do I have that they show up? What's the proportion of data subsets in which they show up? So a dark blue line is above 50%. So 1, 2, 3, 4, 5, 6, 7, 8. I've got 8 markers that show up more than 50% of the time. Everything else, these little blue guys, those are more than 5%, but less than 50%. There's a lot of those. And then there's a lot of stuff that shows up one time and never shows up again. So if you've done your analysis on the 100% of your data set, and you publish that, which we've done, and you say, these are the QTL, well, I could take your same data set, I could probably find another 1,000 QTL if I just keep reanalyzing your data. So the point here is that it's pretty complex, and that's not the end of it. What we also did was we took each family separately. So we just take the families apart, analyze them one at a time, and you ask the question, how often do you find a QTL in this one family? And so here's the B73 by B97 family. Okay, it looks like we have more or less something going on here on chromosome 1, and maybe a few other things. We don't have much power here. We know that's an issue. But basically, this position looks like it shows up in one other family. And this is something we see really commonly, that you can find QTL that are population specific, and you're not going to see them in some other population. So what if they explain 5% of the variation in this one cross? They probably aren't even segregating in some other cross. So how is that information used for breeding? Probably not. Okay, one other thing, if I have time. This is one more example of trying to get the point across that even what we think as relatively simple traits, at least traits that we can measure with high heritability, are actually quite complicated. This is a story where Arnold Hollauer at Iowa State took a set of accessions of Toussaint, which is a lowland tropical race, and took them to Iowa. So that's exotic germplasm. And photoperiod's all wrong. They flower way, way too late. But what he did was he planted basically an isolation field of 10,000 plants and would walk through it every day and flag the earliest 500 plants that flowered. So there was very large population size, very strong selection pressure, but big effective population size. So drift is not a big problem here. And he did that for 10 generations. Okay, and here's an analysis we did on this side of the graph here, where here's the initial cycle, and actually the population size here is the sample is a little low because a lot of them didn't germinate, but as we go through the 10th cycle of selection, you can see the distribution of the population just shift very quickly in terms of earlier flowering, which is what he was selected for. So you see this dramatic response to selection. And actually, one of the reviews on this paper said, has to be simple, must be a major gene, all right? All right, so we throw 45,000 SNP markers at it. And so not only do we have the things phenotyped, we have the same things all genotyped, and we can do population genetic analyses on these. We can also do GWAS because we have individual lines in the field from this thing sampled, and we have them genotyped. And the first thing I'll tell you is when we do the GWAS, when we put the population structure correction in, guess how many things we find that are significant? Maybe 2, right? Maybe 2. It depends. We argue about what's our FDR. 20% FDR, we can maybe find 5, 10, something like that. It's not many. So again, this argues, yeah, it's single gene. It's very simple. If you do the population genetic study where you ask the question, is there a change in allele frequency at this SNP that's bigger than can be explained by drift, you basically come up with thousands and thousands of SNPs. And they're not all clustered, they're all over the genome. Okay, essentially the entire genome is under selection, which means there's lots of genes, and they probably have lots of small effects. And Arnold Hollauer put very strong selection pressure in a big population, and you can actually make pretty substantial gain doing that. Okay, oh, and that's not published yet, we're working on it. All right, so that's kind of that's a couple ways you can look at structured families for mapping. You can go into these recurrent selection populations. You can do these sorts of analyses. Probably the most common thing people do is just association analysis, where you take a sample of, let's say, lines from a breeding program or from a germplasm bank. You put them in the field, you evaluate them, you run a bunch of markers on them, you do what they call a genome-wide association study, and you publish some papers. And we have done this. And the advantage to these diversity panels is you're sampling much greater diversity. That's great. You sample more alleles. The resolution can be much higher, particularly in maize, because the linkage disequilibrium breaks down very fast in diverse material. You don't have to spend all these years making inbred lines out of crosses, which is a lot of work. However, there's some penalties to it. You have to deal with the population substructure. To avoid identifying SNPs being associated with your trait, due to the fact that they happen to be specific to a subpopulation that has some particular value for that trait, which is the population structure false positive effect. So what I'll show you what we have to do is we have to statistically account for that population structure so that you're not picking up those effects and thinking that you've got a causal variation or you're very close to a causal variation. The other problem you can have is you have lots of low frequency alleles. If you just look at the SNP frequencies in these diverse panels, the vast majority of SNPs are very rare. They occur in maybe just a few lines, particularly if you go into exotic germplasm. And the problem there is there's a lot of diversity that's very interesting, but it's very specific to one or two lines, which means you have no statistical power to identify those things in these sample sizes. And then this last thing, and Renee has made this point very well, and I'm glad she did, you have to deal with adaptation somehow. So if you're going to look at some kernel-associated phenotype, and you're growing exotic germplasm in North Carolina or Iowa, and it doesn't flower, okay, so we can identify all the alleles involved in not making seed, but I don't think it really means anything. It just means we're evaluating these things in the wrong environment. So you have to deal with these things, and a lot of what we've done is try to just look at traits where we think maybe we can pretend we don't have to deal with adaptation. Okay, so we got to correct for population structure, and I showed you we did that nested association mapping study. That's easy. We know the family structure because we made the families, and you can just put it as a fixed effect in the model. However, if you get to these diversity panels, it's very complex. You have many layers of recent and deep relationships among lines. Let me just go back and show you this guy. This is a sample of all the inbred lines in the USDA seed bank, and this is just two principal components, but you have this. It's not, you can see clear subgroups, but you have all this stuff that's kind of intermixed, and you don't know what to call it. Is it stiff stock? Is it non-stiff stock? And it's mixed with tropical, and it's, that sort of thing. It's very, very deep layers of relationships. So the most effective way to deal with that is to measure the pairwise genetic similarity for all the lines. And then we'll include a random polygenic background term that says the covariance structure of my genetic background effects is proportional to this genomic relationship matrix. Okay, and I'm actually going to show you an equation where we do that. And then the idea is, so you've got this background genetic variation term that reflects how the material in your sample are related to each other. And then what the GWAS is doing is saying, when you take that part of the variation away, whatever variation is left over, can we explain that variation with the differences in the alleles at a particular SNP? Okay, so going back here. So the phenotypic similarity between individuals is mostly based on the proportion of shared alleles, right? Fisher wrote this paper 98 years ago, and we think he's right. So that if you take two clones or identical twins, phenotypically they look very, very similar, okay? And you know this in your family. And then if you go to full sibs, you can say, yeah, that's a family. You can recognize a family, but there is some variation. They're not all identical. So it kind of look like my brothers, but not exactly. And then if you go to first cousins, it's a little bit harder to tell. Probably you can't tell that they're closely related. And you get more variation within the family. Okay, so we all know that. That's what everything on classical quantitative genetics is based on. The pedigree expectations, half sibs are, have a genetic correlation of 0.25, full sibs, 0.5, this sort of stuff. But those pedigree relationships are expectations. Oh, also, with that information, the animal breeders, they've run on that for, 50 years, and it worked really well. Given pedigree information, you can do pedigree-based predictions. And it works. But those pedigree relationships, they're expectations, or they're averages over all the possible outcomes. Any specific pair of relatives is going to vary around that. So me and my brothers, every pair of us are not exactly correlated additively by 0.5. Some of us are probably 0.45 and some of us are 0.55. Okay? There's variation within that family around that. And what we can do is if you have genome-wide markers, you can estimate the realized genomic relationships among those things. So that's exciting. So here's just a simple example. There's a million examples of this. I just took one of the marker data from one of these biparental families from the nested association mapping population, and I asked for each line what proportion of the alleles came from B73, which is one of the parents. And we expect it should be 0.5 based on pedigree. And the mean is 50.1%. Fisher was right, amazingly, without any marker data. But there's this huge variation around that. And it's this variability around those expectations is where the realized genomic relationships and that genome-wide markers become really powerful. Okay, here's what we do. We're going to do a GWAS. And when we do the GWAS, here's our model up here. And it's, you know, the equation's not that hard. This is the part we're interested in the GWAS. This is saying I'm looking at, I code my SNP that I'm interested in. I have some individuals, I call it 0. They have one allele. The other individuals, let's say they get a 1. They have the other allele. And I'm looking for differences at that marker. Oh, if I can get my arrow back. Okay, what's this thing? This part is the mixed model part. This U is the effect of each line in my sample. So here's a case where we had 279 maize inbreds from all over the world. You just take a bunch of marker data, you estimate how related they are. And so what this is a heat map showing with colors how strong the relationships are. So when you get down to like white, it's basically 0 relationship. They're unrelated. If you have things that are kind of hot red, those things are related. And I didn't order, I think they're alphabetically ordered. And so there's some group here, I don't even know what it is, but these guys, this part of the matrix, these guys are all related. So each row and each column is one of the individuals in your sample. And you're saying, if I look at the variability in my trait, if the differences between any pair of individuals fits what I predicted in terms of the proportion of this matrix, that's background effects. So the background effect gets absorbed in this variance component. So these background effects are random effects, and they have a variance covariance matrix proportional to the realized relationship matrix. And I may have just lost a bunch of people there. This is really important. So I'm going to go back over it again. So okay, but the point here is what we don't care about that part. We're like removing it. It's like a residual part of the model. We want to get rid of it. And what we really care about, right, is we want to find that SNP so we can publish a paper and get our PhD and get out of grad school. That's this part of the model, right? That's trying to hit that, you know, ZMCCT or that disease resistance gene. Okay, so our interest in the GWAS is the SNP effect betas. Now we're going to move into genomic prediction. I'm making a little shift here, and I haven't warned you, but here it comes. And the reason it happens right now is because it's almost exactly the same statistical model, right? So we have some vector of phenotypes. We have some mu. That's the same thing. We have some residual we can't explain. The ZU part, oh my gosh, it's exactly the same thing we do in GWAS. What you have is a set, you have this matrix, so we have 279 lines in the rows and in the columns, and this little cell here is telling me how many alleles, what proportion of the alleles are shared between individual 1 and individual 2. What proportion of the alleles are shared between individual 1 and individual 279? That's this piece of the box over here. And again, you're asking the question, are the phenotypic differences that I see, are they proportional to the amount of the genome that's shared? So if I see that, oh yeah, they share a lot of the genome, and oh yeah, they look the same, that's polygenic background. If I see that, well, they share a lot of the genome, but actually they don't look that similar, that's not polygenic background. So that's either going into residual, or it's something you maybe can explain in the SNP effects. They're, again, almost exactly the same genetic model. And an aside, you'll see there's lots of ways to do genomic prediction. This is not the only one. It's actually equivalent to some other ways that look more complicated. But this thing works. GWAS and genomic prediction are very similar models, but the objectives are completely different. In one case, you're trying to identify specific genes or variants that cause an important effect on your trait. And you want to understand, you want to do, like I'm saying, genetic analysis. Let's find out what the genes are. And if they're big enough effects, maybe we can then use those in breeding. The other part of the model, or the genomic prediction model, is saying, I don't care about any one individual effect. I think my trait is mostly polygenic. Any one SNP much importance as any other SNP. It's totally democratic. It's a vote, right? And so I'm going to predict a line somewhere in this matrix where I don't have any phenotypes. It should be similar to the guys it's related to. That's all that genomic prediction is. You say, I have all these relationships. On some of them I have phenotype data, and so I can measure how well that phenotype similarity matches the genomic similarity, and then I predict. And I predict that they're basically, if they're genetically related, they're going to have similar phenotypes. Something like that. I mean, the details are really complicated, but that's the basic idea. So let's talk a little bit about genomic selection or genomic prediction, because if we get into this world, like I told you, it looks like a lot of traits have many genes, each with small effect. It's really the polygenic model, and it seems like genomic selection is going to be a more appropriate way to use genome-wide markers than doing GWAS. Okay, so it's already been shown, lots of papers on this. It works well when the training data set is closely related to the prediction population. So in other words, you have some subset of your data where you have markers and you have phenotypes. You build a prediction model. You say, what function of my markers predicts the phenotypes? And typically it's simply the more alleles they share, the more similar they're going to be for the phenotype. Then you have another set of your data where you have no phenotype data. So maybe this is a case where you have early generation materials. Some subset of them have been phenotyped, but the phenotyping is expensive, and supposedly markers are really cheap, and in some cases they are. So maybe I can just take that marker data, build the model where I have all the data, and predict the cases where I don't have the phenotype data. And it works well if those two sets of materials are closely related. And the Pioneer people can tell you, I don't know anything about this, but they're doing this. But the trick is, you have to have the training data set closely related to your the things you're applying the model on. Okay? You need narrow genetic space. You would like to have long linkage disequilibrium blocks. So what this means when we go to more diverse material is that, oops, we don't have narrow genetic space. We have LD breaking down very quickly in Maíz, and that causes problems for these predictions if your training set and the prediction set are not closely related. Here's an example from the NAM where you take each biparental family separately, you measure plant height. Heritability is like, I don't know, it's like 90%. It's really high. But you build a prediction model on one family, and you try to predict on another family. And what you see in this graph is that in general, like you get down to these white squares here, there's basically no prediction ability that goes across families in these cases. There's a few cases where you can get up into the 30%. Like in this case, it's a sweet corn family being compared to another sweet corn family. But in general, it does not work very well. It can get worse. Here's a paper out of the CIMMIT group where they took lines from lots of different CIMMIT programs, built a genetic prediction model, and then tried to predict new biparental families, and the prediction correlations between what they predicted and what they actually measured ranged from negative 0.26 to positive 0.29. The average was actually negative. This means somebody did a lot of work to collect marker data, collect phenotype data, and in this case, if you had used that genomic prediction model, on average, you would have been worse off than by selecting it completely at random. Okay, so you're spending money in this case to do something worse than, like my kid could do, picking seed packing. My kid would do a good job, better than me. But a monkey or something like that. Okay? So the training sets have to be closely related to the prediction sets. And this is really a problem, because if you get into diverse genetic material, if we talk about 50 races of maize, each of those races, there's hundreds of accessions. You get into each one of these, they're all like separate cases. And a prediction model in any one case is not going to do anything for another case. So this is hard. Maybe I should, how much time do I have? Should I quit? Keep going? Keep beating this poor thing. So I didn't want to completely give up. That's all the bad news. So here's okay, I'm going to try to get something out of this GWAS type model. What we thought was, okay, maybe most of the genetic architecture is lots of genes with small effects, but maybe you have, in some cases, a few genes with slightly larger effects. They're not big effects, but maybe just big enough where maybe they would account for more than that just genetic background. So what we did was we took our this NAM data, and we did a thing where this cross-validation type stuff, which is just a way to see if this is going to work in reality. So you take the data you have, and then pretend you don't have phenotype data on some of it. So you split your data into a training set. 80% of the data goes into a training set. You hold out 20% here, and you're going to try to build a prediction model on this material and try to predict this test set over here. So what we did was we basically did every time in the training set we redid the GWAS analysis, we identified some markers, we tried to estimate their effects, and we put them into the prediction model in addition to the polygenic background term. And we said, okay, we're going to compare not doing that, which is you just do one analysis on the 80% and you just do the prediction model, versus you do a GWAS and then try to fit those effects in and then compare those things. So can we pick out any signals of association using GWAS and improve our prediction that we could do with the polygenic model? We started with a simulation study because you want to publish, you got to show some simulation. So my student Yang Bian did this simulation where he said, okay, let's take the easy case, 10 loci, each of which explains 3% of the variance. And then, so there's a part that's oligogenic, and then there's another part that's polygenic. And we do this. And what happens is, here's our GBLUT model. Here's the prediction accuracy. It's like 33%. If we do the GWAS, and here's our threshold for declaring a significant result, we greatly increase the predicted accuracy. So if you have a few genes and they have at least a little bit big effects, you may be able to pick them. You may be able to hit them with GWAS and put them into your model, and you can do some real good improving the prediction models. So you get this boost in accuracy. Here's the other part of the simulation. What happens if we make a really polygenic model? So you have a random background term, and then you have 100 loci, each of which explains 0.3% of the variance. Then what happens is, here's the GBLUT model prediction. It's good because it is a polygenic. The model fits the genetic architecture. And here's what happens when you start trying to fit GWAS associations into it. You don't get any gain. In fact, you could do much worse. So the bottom line is, you want to use GWAS in the case where you can find some significant effects. Now, you don't know this before you maybe do the study, but the idea is, go ahead, do the study, do the GWAS scan, and if you find some significant associations, so here's southern leaf blight, so now we're into real data, so we did it with southern leaf blight, with gray leaf spot, and on average, in each subsampling, we picked up either 21 or 14 associations. When you can pick up things at reasonable stringency, they might be big enough to improve your model, and you get a little boost in prediction. So here we went from like 50% to 53%. I don't know. I don't know if this is worth it or not. But all it is data analysis, so you're not doing any more work. The problem is when you get into something like plant height. And I know plant height is a component, but actually plant height is incredibly complex itself. So when you get into these more difficult traits, which are, based on these component traits that all interact together, each of those components might be very complex. So here's plant height. You rarely can find anything with the GWAS. That means the polygenic part is good, and that means fitting the GWAS associations don't do anything, and so there's no point in doing it. It's all polygenic. Okay? So here's the summary. I just want to say everything I've said here is very maize specific, and maize is, I think, quite different from wheat and other self-pollinating species. So if you're a wheat person, you might be able to ignore most of this. Yeah. So that's what I've learned. It's taken me 10 years or 20, something like that. It's hilarious to put this up, right? But really, 10 years ago, I thought, well, complex traits are complex, but again, we can break them down. We can figure out what the major genes are, and we're gonna take complex traits and simplify them with genetics. So basically, that has happened in some special cases, but the large majority of the cases, the complex traits just are complex, and there's no way around it. Okay. The polygenic model often explains most of the observed variation for most of the traits, not all of them, like the tar spot examples and maybe MLN. There may be cases where you can find major gene effects, and that's great. If you can find them, use them. What we've also found, I haven't really talked about this, but if you start really doing some statistics on these things very carefully, you get very scared very quickly, especially if you're like me and you've published a bunch of these papers. And then you have a student who says, well, what happens when we resample the data? And then, So there's a lot of things that, again, you'll find an association in one analysis, the same data, the same environments, the same material, except you did a little resampling on them, and boom, that thing is gone. It disappears. That's very scary. So don't believe your own results, I guess is what I've learned. So the polygenic model looks like it has a lot of promise for genomic prediction, and industry is, they're really going to town with this thing. But the thing, it doesn't solve our problem when we get into diverse genetic material, because the prediction models will break down if you don't have a training set very closely related to your testing set. Why this gets into some deep parts of the genetic architecture, and we actually don't know why this is. It may simply be that allele frequency differences between a training set and a test set are that enough, it will mess it up. Trudy McKay will tell you it's all epistasis, so that's why it doesn't work. And she may be right, I don't know. It also could just be that the LD patterns, the linkage disequilibrium patterns are different. So there's a lot of reasons. And we're probably going to spend the next 10 years doing academic studies on why this thing doesn't work, which isn't going to really help anybody. So we still are left with this question: can we identify causal genes of some larger effect that's going to help in prediction in more diverse germplasm? Maybe is the answer. And there are some cases. I just want to really emphasize that so far those cases have been the special cases. One last thing is that the people who really want to believe in large effect things will say, you have missed all the rare alleles with large effects. And we know this is true, there are rare alleles with large effects, and you cannot pick them up in a GWAS because they're rare. Okay, and that's true. I doubt it's the case for most of these traits, but it is possible that there are, again, these things that happen so rarely that we miss them. And if you do something like a biparental population, you may pick them up. All right, so for the Spanish speakers, I know I talked a lot, and it was very fast, and it was bad, hard to understand. So here we go. Just to be sure I'm really clear, I wrote this slide this morning. This is the summary. You didn't have to listen to anything else. Here it is all. Okay, es difícil identificar los genes que causar variación en características complejas en maíz porque hay demasiados genes y tienen efectos muy pequeños, muy chicos. Okay? También significa que no existe un set poquito de marcadores que son útiles para selección, y es porque un grupo chico de marcadores no puede explicar mucho de la variación y probablemente son específicos para una población. ¿Ok? Se puede usar marcadores distribuidos por toda la genoma para hacer predicción genómica de características complejas. Sí, ese funciona, pero la predicción genómica solo funciona bien cuando el modelo es hecho con una población bien relacionada con la población en que quieres hacer la selección. Okay? I think I'll stop there. I have one more slide on some landrace stuff, but it's already too much. People want to eat lunch. Basta ya, ¿no? Bueno, gracias a la gente que trabaja conmigo, a un montón de gente que trabaja muy duro para finalmente saber que no hay genes de efectos grandes, es muy deprimidos, pero gracias a todo eso y los grupos que nos das dinero para hacer los experimentos. Termino.
Agradecemos al doctor James Holland.
¿Hay espacio para una pregunta? ¿Alguien?
Thank you for the presentation. I just wonder, so yeah, the complex traits, they have a lot of polygenic background and also a lot of interactions possibly would interfere these models. Like how can we manage the G/E interactions when we want to maintain the accuracy of the genomic selection?
Yeah, so right, I haven't even talked about that. Everything we've done so far, the training data sets have been the same environments as the test sets, the validation sets. All you have to do is put your new population in a new environment and your prediction accuracies drop very quickly. If there is G/E for the trait, which there is for almost everything. So, yeah, I mean, this is the other thing. Your training set has to be related to where you're going to apply it both in terms of genetics, which is really what I talked about, but the other part is the environment. And that's very clear. So you probably have to do, again, you have to know your environments. You have to know where what the target set of environments is. Or any predictions you do for some specific environment aren't going to work. So that's very clear. Yeah, you better worry about it. So more data, good data. Everything Renee said was exactly right. You need good data, right? And you need to be in the right environments. And if you're not, you can sequence all you want, and it's not going to help.
Le hacemos entrega de un pequeño obsequio y la constancia de su participación en este evento.